Alright guys, so today what we're going to do is we're going to learn how to do what's known as a dihybrid cross. Up to this point we've only been doing monohybrid crosses, so we've only been looking at one trait, like hair color, and it's either brown hair or blonde hair, eye color, and it's either blue or yellow or something like that. Now let's look at two traits simultaneously. So the one we're going to do as an example is I just made some stuff up about guinea pigs. And I'm going to read you the problem because we're still going to follow the same protocol. So we still want to read the problem carefully and figure out what exactly it is we're trying to know, what we're trying to figure out, and what kind of information they've given us. So it says guinea pigs can be either brown, big B, or yellow, little b. So obviously the brown fur color is dominant. And they can have ear flaps. So that would be like having like an external ear, kind of like we do, right? Or not. E. So they could have ear flaps like we do or no ear flaps. This is completely made up stuff, all right? So that's kind of our information that we have. Those are our two traits. You can either be brown or yellow, or you can have ears or not. Okay, so we're going to look at both of these simultaneously. So it says, if a heterozygous, brown, earless guinea pig was mated to a yellow, homozygous dominant ear-having guinea pig, what would the resultant phenotypic ratio be? All right, so obviously, I'm going to mate a heterozygous brown earless with a yellow homozygous ear having. So those are my two guinea pigs, and I want to know what the phenotypic ratio is going to be. That's what I'm eventually trying to find. So I know I've got this guy and this girl, I guess, and then here would be our phenotypic ratio. That's what we're trying to find. So first thing I'm going to do to make my life easier is I'm going to make both keys so I have them available. Okay. So you got to double up because it's two traits, so now we got to do two keys. So you're going to do big B, big B, big B, little b, little b, little b, and this is going to be brown, and this is going to be brown, and this is going to be yellow. And your second key, we're going to do big E, little e, and little e, little e. I like that, big E, big E. It sounds like I'm saying Big E, Notorious B.I.G. All right, so ears, and no ears. All right, so there's our two keys. We got the problem. We know exactly what we're supposed to be solving for. So let's write out our cross. So let's figure out what would this individual look like. Heterozygous, brown, and earless. Well, there's only one way to be brown and heterozygous. That's big B, little b. So our first individual, we know it's going to be big B, little b. So heterozygous, brown, and they're earless. Well, there's only one way to not have ears. That's little e, little e. So as you can see, we've got one trait and a second trait, hence dihybrid, 2. All right, so that's our one individual. And we're going to cross this individual with some individual that is yellow and homozygous dominant for having ears. There's only one way to be yellow, obviously. So they're going to be little b. And now it says homozygous dominant ear having. All right, so it can either be this or this. Well, obviously that's dominant, so it's going to be big E, big E. All right, so far this is the easy stuff. Now, what we have to do, and this is where kids tend to fall down, is we have to figure out the gametes, the stuff that goes on the left of the Punnett square and the top of the Punnett square. And with dihybrids, it's a little bit trickier than it is with monohybrids because monohybrids just separate stuff. But what we have to do is, if you remember back when we talked about some genetic stuff, there's this idea of segregation. So what we're saying is these two B's, right, we have to separate these guys from one another, and we have to separate these guys from one another. And we can do any possible combination of how these guys are going to get together. That's the genetic variation. That's that independent assortment, right? All these genes can do their own thing. Now, the easiest way that I've found to figure this out is to do something that you guys have done in math class for years. FOIL. And you use the FOIL method when you guys are trying to figure out like factoring and that kind of stuff. And if you remember, it's first, oops, helps if I can spell, outer, inner, and last. That's supposed to be a U there. So what we're going to do to find our gametes. First thing we're going to do, zoom this in a little bit. Whoops, going the wrong way here. Is I'm going to take a look at this. And I want to put, if we put parentheses around this, if we just pretend this is math class, okay? And these are our different pieces of information that we're trying to factor. I want to put the first two things together. All right, so that's going to be big B, big E. And then I'm going to take the outer ones, which means I'm going to do big B and again, little e, right? The only possible thing that I can put this big B with is that little e and that little e, but I got to put it with both of them. So there's going to be my first two and then my outer two. Now I got to go to the inner two. Well, that means I got to take this little b, and these two are next to each other. This little b and little e. I'm going to put them together, and I'm going to put the last two, this one and this one, together. And what this means is that we're going to end up with more gametes. And that means our Punnett square is going to end up being larger. Our Punnett square is not going to be 4 squares. It's going to be 16. All right, so using this FOIL method, I'm going to go down the left-hand side of this. I'm going to figure out what my gametes are going to be. So I'm going to put the first two things together. I'm going to do B and E. All right, so I'm going to do big B and little e. That's one of my gametes. And I got to do the outer two, which means I got to go with this big B and this little e here. Now, yes, some of these are going to look similar. All right, I'm going to do little b, little e, and little b, little e. I got to do the same thing with this guy over here. Again, I'm going to take the first two and put them together. Now, obviously, you can kind of see what's going to happen across the top. They should all end up being exactly alike. So, all right, I put the first two, put the outer two, I'm going to put the inner two together, and I'm going to put the last two together, this one and this one. And so they're all going to be BE. Now, here's the thing, guys. If you don't have two letters above each of your columns and next to each of your rows, you didn't figure out your gametes correctly, and you're probably going to get the whole thing wrong. So this is a critical step. This part right here, like I said, is the part where kids generally tend to make the mistake. They don't find the gametes correctly, right, to go across the top and the side, and then they get the wrong stuff in here, and then they can't answer the question. So this part right here is the most important. If you have questions or if you have trouble, if you don't understand, email me. We can do a tutoring session and I can walk you through it. We can do another couple of problems or something like that. Now, all I got to do now, right, since I've got all my letters, is I just drag everything in. Now, what I like to do is I always like to keep my dominants first, and then I keep my recessive second, and I'm going to keep everything in the same order I was writing it up here. So I'm going to do all my B's first. Now, for me, it's usually easier to do one letter at a time. I kind of get into a rhythm of filling it out, and it makes my life a little easier. All right, now we can go back and do the E's. So I'm going to drag everything in. So you can see, right, my resultant genotype is going to have 4 letters, which makes sense. The parents had 4 letters, so all the offspring should as well. I'm going to be big E, little e, big E, little e, big E, little e, big E, little e. Do-do-do-do-do. Whoops. Big E, little. These are all big E, little e. All right, so the Punnett square has gotten bigger. We've got 2 different traits we're dealing with. But as far as understanding what we're trying to figure out, now it just wants to know the phenotypic ratio. Remember what a phenotype is. It's what do these things look like. So we have to come back to our keys and kind of figure out what does everything look like in here. Well, we only have 4 possibilities. You can be brown with ears, brown with no ears, yellow with ears, or yellow with no ears. That's it. There's only 4 possibilities. So it looks like these 2 columns, or these 2 rows going across right here, these guys are brown with ears. All right, so we got 1, 2, 3, 4, 5, 6, 7. We got 8 of them that tend to be brown with ears. And then these guys are yellow with ears, so 8. So our ratio is going to be 8 to 8, or you can reduce it if you would like. 1 brown with ears, 2, 1 yellow with ears. If I asked you percentages, you would tell me 50% yellow with ears, 50% brown with ears, right? So all these guys are going to be brown with ears. All these guys are going to be yellow with ears. It looks like every single one of them is going to be heterozygous for ears. It looks like we're going to be heterozygous for the brown color, and obviously if you're going to be yellow, you have to be recessive. All right. So this is the example problem, right? I want you to use this methodology to try to help you go through some of the problems I'm going to give you. It's going to be SpongeBob. It's going to be a PDF style like it's been before. So it should look familiar. It's going to kind of start you out with some baby steps and then walk you through this method. If you have trouble, email, give me a shout. We can go online, do some video conferencing, tutoring, that kind of stuff. All right, guys. Have a good one. I hope you understand this. Like I said, get a hold of me if you need help.